Okay, I forgot to get the sweetener this morning, but does anybody need a coffee that doesn't have one? I think she beat you. Gosh, guys in this class. There you go. And how about a bowl of used Brussels sprouts from dinner last night? You're still all right. Pass that pumpkin muffin back to the winner. Thanks very much. Was anybody here out at the demonstrations against the cuts in public education yesterday? Yeah? Great. Thanks very much. The future of public education is seriously at risk, as is the entire future of the University of California. And since California really sets the model for much of the public university and public school systems in the country, it's really important that we do everything we can to defend it. So thanks very much. I want to remind you I will not be holding office hours today. I have to get on a plane to San Diego almost immediately for a meeting there. I held one yesterday. I'll try to hold an extra one next week. Monday we'll be back on the regular schedule. What I want to do for most of our time today is to talk about the sexual life cycle that's enjoyed by so many organisms on the planet. But to be honest, the asexual lifestyle is incredibly successful. And let's spend a little bit of time talking about the successes of the asexual lifestyle. And I'm not preaching abstinence here. These are just organisms that don't happen to use gametes to make a living. So Hydra is an example that you probably saw in your high school biology class. And Hydra are animals that reproduce by asexual budding. They simply grow another Hydra off of their side. It would be rather inconvenient for us if we were to reproduce this way, and be difficult on our fashion sense as well. So you can have something as complicated as an animal with multiple different cell types reproducing asexually. The yeast that you've dealt with in lab already and that we've already discussed have a mode of reproduction called asexual reproduction, the budding. We'll talk about their sexual cycle later. But many different fungi reproduce simply asexually by budding. Now, something that may surprise you is that this creature here, which is a really lousy picture of a dandelion, reproduces asexually. You're probably thinking, he must be nuts. I've seen dandelion seed all over the place. Yes, you have. But curiously, dandelions produce seeds not through meiosis, but through a special mitotic division and development. So the seeds that are on the top of a dandelion are actually asexually produced seeds. They're not haploid. They're seeds that are diploid seeds produced asexually, in a process that's known as apomixis, the ability to make seed, diploid seed, without going through meiosis. This is also seen in a weed called Heraceium that will have a tragic impact on our story that we'll discuss on Monday. Be prepared for that. So those are some of the successes of asexual reproduction, but mostly what we'll talk about today is meiosis, which is a specialized cell division that reduces the ploidy of cells from an organism by 50% so that fusion between the gametes produced by meiosis can restore the normal ploidy. Now notice that I didn't say diploidy, because meiosis applies to many different kinds of ploidy. So diploid animals and plants through meiosis produce haploid gametes. Tetraploid plants and animals produce diploid gametes. So watermelons, for example, are tetraploid, and there's no shortage of seed produced by watermelon. Hexaploid species produce triploid gametes. Now, I mentioned before that meiosis in a triploid is a bit problematic, and we'll demonstrate that today with the triploid banana. Would you take a piece off the end of the banana? Okay, now let's look really hard into that banana. And do you see anything that looks like seed?
Not really.
She says she doesn't see anything that looks like seeds. Has anybody ever seen anything that looks like a seed in a banana? Yeah, have you seen it along the midsection, those tiny little white, stubby little things? Yeah, they are aborted seeds that are aborted. You can eat the banana if you like. You want the whole thing? Yeah, okay, here. You can eat bananas quite safely because all of those seeds are aborted soft little ghost seeds because of the hell that breaks loose when a triploid tries to go through meiosis. And as you'll see when we discuss the mechanism, there's no simple, regular segregation to make sure that all of the seeds get the right number of all of the genes, and so they suffer from massive amounts of gene dosage effects which cause the seeds to die. Now, interestingly, that banana, and probably every banana that you've ever eaten except for the exotic ones at Berkeley Bowl, are of a particular strain of banana called the Cavendish. Chiquita and all the major producers of bananas are all using the Cavendish, and there is a fungal pathogen that is wreaking havoc among all Cavendish bananas. So we have to breed into bananas resistance, which they have lost, and just think about how difficult banana breeding must be for a triploid organism that produces invariably dead seeds. There's a wonderful article in the New Yorker recently about a research laboratory in Costa Rica, the only one in the world that does banana genetics, and the article describes the challenges of trying to breed resistance into a triploid organism. Now, where did meiosis come from? Well, on BSpace, I've posted a PDF of an article by Robin Holliday, which discusses his views about how evolution evolved. And one of the drivers for the evolution of meiosis is something that was first proposed by Hermann Muller, and it's been referred to as Muller's ratchet. And Muller's ratchet simply says that any organism that grows exclusively asexually will accumulate mutations. Most of those will be bad. They'll continue to pass those mutations on to subsequent generations, which will again accumulate mutations. And if you haven't got a way of dealing out all of the deuces in your deck, eventually you end up losing all your face cards. You end up being genetically compromised. So that's the thought about what really drives meiosis. But I have to say there are some organisms out there that really make you scratch your head, including a class of single-celled organisms called rotifers, and particularly the deloid rotifers. By studying the evolution of different species of rotifers, it's been possible to deduce that the deloid rotifers came from earlier sexually reproducing rotifers, but something like 100 million years ago, they gave up on sex, yet they still exist, and they seem to be able to avoid the disaster of Muller's ratchet. So there's a lot about meiosis that we still don't know, but on balance, sexual reproduction seems to be relatively successful and relatively enjoyable. So the sexual lifestyle actually varies considerably among organisms on the planet, and we'll talk about both humans and plants. So most of our tissue, of course, is somatic tissue, but the production of gametes by meiosis occurs only in specialized organs, the gonads. The gonads, in the case of diploids like us, produce gametes that are haploid, 1N. And in the situation with humans, our haploid cells don't really have, haploid gametes are unable to divide on their own and have a life that is short unless they manage to fuse, fertilization, to reestablish the diploid phase. Okay? Then the diploid cells divide, developmental biology happens, and you restore us. So in human, there isn't a lot of time for the impact of mutations through Muller's ratchet to get weeded out. It is just really during this short-lived haploid gamete phase. But some plants have a very interesting specialized way of dealing with haploidy and diploidy. In plants, haploid cells produced by meiosis are capable of creating what's called the gametophyte generation. And it's a bunch of tissue, plant tissue. It looks like a plant in which all of the cells are haploid. And it's in this gametophyte generation that the impact of selection on those mutations which may have occurred can either select for or against the growth of particular gametophytes of particular gene sets. So the gametophyte generation then also gives rise to gametes that will fuse to produce the diploid phase of the plant life cycle. This is described in greater detail for you in the book. Now, one thing that is particularly interesting about human reproduction, and especially the development of the female gametes, is how early it happens. So the precursors for your eggs, you of the female sex, were set aside very early in your development, while you were still a fetus. That's when all of the precursor oocytes that you're ever going to have were created and set aside. That's all you've got for your entire life. And once you reach fertility, one of those matures per month on average. Now, what's interesting is that those ogonio precursors in humans are actually arrested in meiosis I. So they are stored in the first meiotic division, and they complete that meiotic division one cell at a time, once per month, post-fertility rather. Now, what's very, very interesting is there have been a number of epidemiological studies that have been done that suggest that the environment in which your oligonial precursor cells developed may actually set the state of the genes in those oocytes such that they will behave differently depending upon that environment later in life. One example of that is the famous Dutch winter of starvation. When the Nazis invaded Holland in World War II, there was a substantial resistance, and the Nazis responded to that by simply trying to starve the entire nation to death. There was one winter when people were getting by eating leather, eating wood. I mean, it was pretty bad. But what's very interesting is that epidemiologists have discovered that women who became pregnant such that their first trimester occurred during the Dutch winter of starvation have given rise to people, to children, in which their genes have been set in the so-called the greedy gene mode. That is, these genes in the offspring from people conceived during this winter are genes that tend to store nutrients much more efficiently than people that were conceived ordinarily. And in fact, there is an entire generation of the Dutch now that are substantially taller than the Dutch ever were before, and for the rest of Europe. Those of you that watch the Dutch soccer team probably have seen the consequences of that. Of course, there are substantial consequences now that we live in an age of excess calories, and the Dutch are actually now suffering from the consequence of having genes set in the greedy mode in a world where calories are now in excess. So that's really a very interesting point that you may be influenced by how well your grandmother took care of herself while your mother was a fetus, while the oocytes that led to you were being developed. So, you know, if you have any particular problems in life, blame your grandmother. Okay. Pretty interesting stuff. The mechanisms by which this work probably involves things called epigenetics. We'll talk something about that later on. Now, because you're using yeast in the lab, let me explain to you that yeast have, in addition to the asexual lifestyle, a sexual lifestyle. And the complementation tests that you'll be doing or are doing in the lab depend upon the sex life of yeast. Yeast come in two sexes. Their sexes are called A and alpha. A cells look like this. Alpha cells look like that. There is not a single person on this planet that can tell an A cell from an alpha cell by looking at one, but they all know. They make pheromones that are specific to A's or alphas, so-called A factor or alpha factor, that are designed to arrest cells of the opposite mating type and prepare them for mating, for fusion. As a result of that fusion, we have a diploid zygote that's made. The diploid zygote that's made produces buds, which are diploid buds. These diploid buds cells can undergo endless numbers of mitotic cycles, but when stimulated in the appropriate way, in this case limiting nitrogen, they undergo meiosis. producing all four products of a single meiosis packaged together conveniently in a sac called an ascus. And it is the ability to study all the products of meiosis at one time that is responsible for our deep understanding of the events that actually happen in meiosis. These haploid spores are of two types, two A's and two alphas. And when they're put back in a rich nutritional environment, they germinate, bud, and reinitiate the life cycle. So even yeast have a sexual lifestyle. And in the lab, you're only going to go through part of this, the mating of haploids to make diploids. You won't be doing an exercise where you take them through meiosis, but you could. Let's pause and see if there's any questions so far. Yes?
Is there a reason why we don't call the two mating types male and female?
The question is, why don't we call the two mating types male and female? It's common in organisms in which there is no sexual dimorphism between the two sexes to not call them male or female, but to call them something else. In a different species of fungi, the two sexes are called plus and minus. Okay? I'm just happy that they weren't called alpha and aleph. I mean, it's confusing enough with A and alpha. Okay. Yes?
Going back to the 3N, last week we talked about the facts, whatever, and we said that the S phase would be kind of like the 3N. So then if
3C. So the question is, when the facts analysis, the intermediate positions between 2C and 4C in the replication of a diploid would be 3C. Okay? Other questions? Okay, great. For our discussion today, and critically for our discussion on Monday and Wednesday next week, I need to introduce some vocabulary to you that you have seen before, but I'm going to take the time to belabor this because if we don't get this down, we're going to have a difficult time communicating effectively with each other about aspects of genetics that are central to the course. So the first vocabulary word that we'll deal with is genotype. You've already encountered this in the lab, and I'll use the definition of genotype here, the particular collection of genes and variants of genes in an individual. Okay, so each of you has a unique genotype. Each fly stock that you used in the lab has a unique genotype that you can read by reading the label on the bottle. So phenotype is related to genotype in that it is the outward appearance of an individual as impacted by its genotype. The challenging thing about the phenotype, though, is that it is also impacted by the environment. So we use conditions in the lab that are extremely well controlled to try to minimize the impact of environment on phenotype. And in the lab, genotype is pretty good at allowing us to predict what the phenotype of an individual will be if we can control environmental variables. The trick, though, is that going in the other direction is really only imperfectly done. Phenotype cannot perfectly predict the genotype. And I'll give you an example here, but I have to warn you that this example depends upon the context into which we describe phenotype. So you're using mutations that affect histidine biosynthesis to learn about the complementation test. And so histidine biosynthesis starts with phosphoribosyl pyrophosphate produced from something else in the cell. And there are 9 steps catalyzed by 9 enzymes with 9 different genes. And mutations in any one of these genes could be described by the same phenotype. His minus for histidine auxotrophy. So that's an example then of the ambiguity of phenotype as to how it relates to genotype. Okay? Now here's the trick, though. Suppose rather than defining our phenotype of interest as histidine prototrophy or histidine auxotrophy, suppose that we said, well, what about indole glycerol phosphate dehydrogenase as my phenotype? Indole glycerol phosphate dehydrogenase is an enzyme encoded by one of these genes, and if that was the phenotype that we were measuring, that would be a closer link between phenotype and genotype and we would be able to predict the genotype of a cell if we were able to see that it lacked that particular enzyme, as if that were our phenotype. So phenotype is contextual. That's the key point that I want you to understand. Genotype is not. Any questions on that? Okay. The next two words we have to deal with are really simple, yet they have, from time to time, created some confusion. A locus is not the same thing as a gene. A locus is simply a position on a chromosome occupied by a gene. So you can think of it as a point on a line. It is just a spot, a position. Okay? What a gene is, I'm going to actually punt on until Monday, because you all know what a gene is, sort of. But on Monday, you'll see that the definition of a gene is a little fuzzy. So we're just going to kind of leave that slightly ambiguous for now. But allele is something we will deal with today, which is the alternate forms of a gene at a particular locus. Okay? So genes can come in different flavors, any given gene. So for example, a particular gene could be of the wild type allele. You've seen wild type in the lab. Or it could be of the mutant allele. Okay? So what's the definition of wild type? Anybody know? What's wild type? Am I wild type? What do you think? So the suggestion was that wild type is the way that things occur most often in nature. That would be a very sensible definition of wild type. In fact, that's not the rigorous definition of wild type. Wild type, believe it or not, is defined utterly arbitrarily. Okay? So an individual geneticist will pick an individual or a pair of individuals from a particular species and declare those the type specimen of that species, and by definition, whatever genes were in those original individuals chosen for study are defined as wild type. Now you're right that since most organisms in the wild sort of look alike, most organisms will have the same form of the gene, and by chance, if you choose an individual, the wild type allele ought to be the most common allele in the population, but it doesn't have to be that way, and in fact is not that way when we get to human genetics. Okay? So it's arbitrary. Now let's consider the gene that makes a protein that you know something about, like beta globin. How many alleles are possible in beta globin? We've talked about wild type, and we've talked about mutant. How many beta globin alleles do you think there are on the planet? Or how many could there be on the planet? What do you think? In high school, you probably heard about hemoglobin, sickle cell hemoglobin. That's the HbS allele. So we've got wild type, we've got HbS. How many alleles could there be of that gene? Well, if the gene is about 1,500 nucleotides long, it's actually bigger, but let's assume that, then each of those nucleotides could be 4 different things. Right? So there's 6,000 possible alleles, or we could have pairs of nucleotides changing at a time. We could have deletions, we could have insertions, and in fact, there are, for all practical purposes, a gajillion different possible alleles at any particular gene. Okay? So that's an important point. So alleles are alternate forms of a gene at a particular locus, and there's lots of different kinds of alleles. The other thing we need to deal with is homozygosity and heterozygosity. So we cannot speak of an individual typically as being a homozygote because homozygosity is defined on a gene-by-gene basis. So homozygous refers to the same allele at the same locus on both chromosomes. And heterozygous refers to different alleles on both chromosomes. So in this diploid, at gene 1, we're homozygous, in this case for the wild-type allele, using the nomenclature that you're familiar with. At gene 2, we're heterozygous. At gene 900, we're heterozygous. Gene 902, we're heterozygous. Gene 903, we're heterozygous. Okay? There are exceptions. Highly inbred organisms like specialized inbred strains of mice can be homozygous at all loci, but typically in the species in the wild, you don't see that. Now, there is another subtlety here, which we won't use much today. We won't use it much on Monday, but on Wednesday we will, and that's the issue of phase. And phase refers to which alleles of different genes are on the same DNA molecule. So with respect to these genes, the wild-type allele of gene 900 and the wild-type allele of gene 902 are not on the same DNA molecule. The mutant gene of 902 and the mutant gene of 903 are on the same molecule. That will turn out to be significant to us because this individual could have the same genotype regardless of which DNA molecule carried which allele. But depending upon the orientation of the alleles and the molecules, the gametes that it will transmit to the next generation will be rather different. We'll come to that point later on. Let me just introduce the concept of phase right now. And the last point is a subtlety that some people have difficulty with. It's really quite simple, and that's the concept of a genetic marker. And a genetic marker is any trait caused by an allele of a gene that can be used to track that allele in a cross. So already in the lab, you've seen examples of this. In the fly lab, white eyes, right, is caused by a mutant allele of the white gene. It creates a trait. It's a genetic marker. That phenotype of the eye can be used to track that allele through a cross. That would be what I would call an indirect example of a genetic marker. You're seeing something far removed from the mutation that actually causes it. The ultimate indirect example is looking at DNA sequence itself. Here's a short stretch of sequence on one chromosome. Here is an allele of that sequence, differs right there. If you're measuring DNA sequence, this difference is a direct allele, a direct genetic marker of that locus. So the concept of genetic marker is simple, but there are a wide range in which we can use genetic markers. Any questions on that? Okay. To warn you, we've discussed in the past the ambiguity about what a line means in introductory biology. Today, a line represents a chromosome and not a strand of DNA, not a strand of RNA, and I'll try to always make that clear by putting a dot on that line to represent the centromere where the kinetochore will be assembled. So what I'm showing you here are, in this diagram, is 4 double-stranded DNA molecules represented as 4 different chromosomes, 2 pairs of homologous chromosomes. Hopefully that'll be clear to you. And what we'll spend the next few boards doing is discussing the events that will happen in mitosis and meiosis following the fertilization of individuals of this genotype. Imagine this is the Indian monkjack with its 3 chromosomes per haploid genome. And we'll have the big A, big B, and big D alleles being contributed by the sperm. The little a, little b, little d alleles contributed by the egg. Notice not to scale. And this fertilization will produce for us this individual, 3 pairs of homologous chromosomes, each pair of homologous chromosomes being heterozygous for genetic markers that we'll refer to as the A, B, and D loci. Any questions so far? Okay. In that case, I need a volunteer. Any volunteers? Great. Come on down. What's your name? Jesus? My man. Okay, what I'm going to ask Jesus to do on this board is the same thing that I want you to do now as a self-test on your notes, and that is to draw a representation of what the first mitotic metaphase will look like for this Indian munkjack fertilized zygote. Okay? So draw that. And Jesus, can I get you to draw this over here? And would you like colored chalk to do that with?
Orange, I don't know.
Well, it's a high-budget show. We've got colors. I would suggest pink and blue myself, but whatever you like, okay? So just take the time to draw metaphase 1 of mitosis. What would it look like? Concentrate really on just the chromosomes and their alignment. Don't worry about the spindle and all that stuff. You got metaphase 1 of mitosis. Nope, I want metaphase of mitosis. Thank you, Mike.
So once this cell divides, right, into...
So let me make sure everybody knows what the goal is. So what I want to, so here's the big issue. The big issue is that you will never understand the difference between mitosis and meiosis unless you understand the difference between the way the chromosomes position themselves at metaphase in the two. So as a reminder, Jesus and you are going to draw metaphase of mitosis of this animal. Okay? So show us what it's going to look like. You guys do the same.
So this is diploid, right?
This is diploid, yep.
So every one of these is going to duplicate and create.
That's right. This is G1. So metaphase is going to come after S phase. And so Jesus is going to show us the sister chromatids for each of these chromosomes produced by replication. Okay? Why don't you give yourself a bigger cell here? Okay? Yeah. Circularity is overrated. Is that your final answer?
Yeah.
All right. Thank you very much. That's exactly right, except the genotype has changed on us. Jesus did exactly the logical thing of using A, B, and C. You will note over here that I use A, B, and D. Oh. So we had a high mutation rate. Conceptually, that's fine. The reason I use D is that it's really hard to tell uppercase C from lowercase c, and that's really the only reason. But thanks very much. Good job. Now, I realize, of course, you guys have already had the same example and asked to do the same thing in lab class, but that's an indication of how important this concept is. So the key thing here is that we have not paired any homologous chromosomes. What we're looking at in each case here are the sister chromatids that are produced by the replication of the chromosome with the big A allele, unpaired with the sister chromatids held together by cohesion of the replication of the chromosome with the little a allele. Okay? Notice that we have an orientation of all the blues up here and all the pinks down here. That's one of multiple different random orientations that we could have had, and you had only time to draw one, so that's perfect. Okay? Any questions about that at all? Now, your text does a great job describing meiosis. Really, one of the best jobs I've seen in any biology text. And so I'm not going to describe all of meiosis for you, but I am going to spend some time dealing with the points that are really the most essential. So in the gametes of this Indian monkjack, once it becomes of age, what we'll do is to look at the orientation of the homologous chromosomes. And here is the key point. What we have is sister chromatids resulting from DNA replication and the pairing of the homologous chromosomes like that, okay? The diagram is really not adequate to show you that those chromosomes are really paired like that. Trust me, they are. I'll prove it to you on the last board, but for now, just understand that they're very tightly paired. I've chosen to put the little a chromosome to the left and the big A chromosome to the right. That was arbitrary. I could have put little a to the right as I've done over here. It doesn't matter who's on top, who's on bottom, who's in the middle. The key point is that they're paired. Homologous chromosome pairing in meiosis 1 is probably the key event that led to the evolution of meiosis. Here's an interesting aside that we may have time to come back to later. Note the monopolar attachment of the kinetochore to the spindle. Right? Here's a kinetochore that is attached to one microtubule. Remember, in mitosis, each kinetochore was attached to two microtubules, and it was the tension that was produced by the chromosome having the opportunity to go both ways that signaled the cell when the metaphase was complete and anaphase was ready to begin. Note here that we do not have that. This will create a question about where does the tension come from on the kinetochore spindle attachment that we'll need to resolve later. Okay. So after homologous pairing, after the chromosomes are attached to the spindle, after the spindle knows that every single chromosome is attached to a microtubule monopolarly, then we get the completion of anaphase 1 and telophase, and resulting in cells here or here, right? And then the second meiotic division, which is really just the mitotic division that we've just discussed, reducing the ploidy to haploidy. Okay? So what's happened here? Well, we've had a really interesting cell cycle. The meiotic cell cycle had an S phase, which doubled the number of chromosomes, right? We want to get down to the haploid cell cycle, the haploid cells, and so what we've had to do, given that original S phase, is to have two different spindle segregation mechanisms occurring, suppressing what would have been another S phase between those two. So you should be thinking about what we learned about cell cycle control last time that could explain how you would evolve this kind of a life cycle. Now, you could argue that meiosis could have been more efficiently done by simply dividing a cell without going through S phase. Yeah, it's true, but that isn't the way it happened. Okay? Apparently, meiosis happened only once because every meiosis in every creature that we know about on the planet does it this way, replicating the genome first, then dividing it twice. So, key points. 1, the genotype of our haploids here are different from the parents. So meiosis is creating genetic diversity. Could this meiosis have produced genotypes of the gametes that were identical to the genotype of the gamete that led to this Indian monkjack? Could that have happened? What do you think? Sure. It could have happened if all of the pinks were on one side and all the blues were on the other, but since it's random, it didn't. So how many different gametes could be produced by this meiosis? How many different genotypes could be produced? How many? What do you think?
Ignoring crossing over 8.
Ignoring crossing over 8, exactly. So meiosis can be a relatively powerful producer of genetic variation in the gametes, especially as the number of chromosomes go up. Has anyone here ever been told that, hey kid, you are 1 in a million or something to that effect? No? Oh man, parental abuse, right? Well, in fact, you're much better than that. Consider this. Each of your chromosomes is different at lots of different positions, and ignoring crossing over, which we'll come to in a second, you have 23 chromosomes. So the number of different genotypes that could be produced by a meiosis with 23 different chromosomes is 2 to the 23rd, so that's 8 million, right? And since you are the product of 2 different meioses, the probability of you ignoring meiosis is 8 million squared, or 64 times 10 to the 12th, okay? So to say that you're 1 in a million is a gross misrepresentation of your true uniqueness. Also, bear in mind that any pair of your homologous chromosomes differ from each other at about 150,000 single base pair positions, okay? So your 2 genomes are really quite different. Any questions on that? No? Okay. Yes?
How does that compare with twins?
Oh, how does that compare with twins? Great question. Identical twins are genetically said to be identical. They come from one egg, one fertilization event, which divided to produce 2 cells, which for some reason that may be understood, but I don't understand it, those 2 cells, rather than becoming 2 cells of one individual, got separated and became 2 cells of 2 individuals. However, now that we have the capacity to sequence genomes, people have started to sequence the genomes of identical twins. And guess what? They're not identical. Yeah, very interesting. And this is, in fact, one of the ways in which we get a sense about what is the rate to which genetic diversity is created in a single generation. Yeah, it's very, very interesting. That some of the non-identities involve things called copy number variants, which I'll probably get to sometime late next week. But they're pretty similar. Non-identical twins, fraternal twins, are genetically no more similar than brother and sister. Or, well, if they're of opposite sexes, right? They're no more different than 2 kids in the same family born different times. Yeah. Other questions? All right. Now I'm going to introduce the coolest thing in sex. Well, shows I don't get out a lot, huh? And that is recombination. So recombination, I'll define as the physical exchange of DNA between chromosomes. Recombination happens all the time in sexually reproducing creatures. And recombination happens on average once per chromosome arm per meiosis. And I'll draw a recombination event here. I'm not going to use the Indian monkjack example down there. I'm going to change the rules here. And note that I've put genetic markers both near the centromere and far from the centromere on these chromosomes. And we have 2 different pairs of homologous chromosomes. So recombination the consequences of recombination can be seen in the microscope by looking at chromosomes from gonadal tissue at the right stage of development. And what you see looks sort of like that. You don't see the letters, but you see a crossover of the chromosome arm. That's referred to as a chiasmata. And that chiasmata marks the position at which the physical exchange between the DNA molecules happened earlier in meiosis. Your text will describe all of the phases of meiosis, and that'll tell you exactly when recombination happens. Now, as a result of this recombination and that recombination, after the first meiotic division, what we see are these 2 cells now, which have some chromatids that are made up of atoms that came from the other chromosome. So you can see that we now have, when this chromosome went to that pole, homozygous for the big A allele, but because there was a crossover event happening, that centromere drags with it one little b and one big B allele. Now, what is difficult to see in such diagrams is that, in fact, there has been a breakage of the blue duplex and a breakage of the pink duplex, and they have been sealed back together now, so that the proper segregation here involves this chromatid as well as zip, 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 the end of that chromatid. It'll be clearer in just a second on the next board. Okay? Now, in the second meiotic division, notice that what we see in the second meiotic division is a bipolar attachment of each kinetochore to the spindle. That's different from meiosis 1. So the requirements for the evolution of meiosis were really driven by homologous pairing. And in the article that I posted on BSpace, Robin Holliday discusses why he thinks that evolved. Recombination at high frequency. The retention of sister chromatid cohesion in meiosis 1. And the suppression of a second S phase in meiosis 2. You put those four things together, and bingo, you've got meiosis. And recombination. Now, that diagram at the top would make you think that the exchange of chromosome arms is a little sloppy. But in fact, recombination results in no loss or gain of DNA at all. And the amazing thing about it, and we still do not understand the miracle by which homologous sequences align. So I'm showing you here the blue sister chromatids and the homologous pink sister chromatids aligning in metaphase 1 of meiosis. They do so through the help of something called the synaptonemal complex, which mediates this pairing. And actually, Abby Dernberg, one of our colleagues here at Berkeley, is one of the world's experts on how the synaptonemal complex brings homologous sequences together. Although she is an expert at it and the world expert at it, there is still much mystery to be understood about how these sequences align with what I'll refer to as atomic precision. Now, here's a quiz for you. How many DNA strands am I representing here? If you don't come up with the answer 4, you're on shaky ground with your understanding of meiosis. So let's zoom in on one of these chiasmata with a super high-powered microscope. And what I'm showing you here is the synaptonemal complex pairing these two sequences. Notice I have an AT here, an AT here, a GC here, a GC here. These sequences are aligned so that exactly the same nucleotides are next to exactly the same nucleotides. What I'm going to do now is to pull a fast one on you, though. I'm going to propagate a fiction that will last until MCB 110 and tell you how we used to think recombination happened. This model is perfectly good. It's kind of like classical mechanics is perfectly good for most things in the physical world, but the real mechanism of recombination is much more interesting. But in this classic model, what we're going to imagine is a hatchet that cleaves this duplex between this A and T and that T and A, and a hatchet that cleaves that duplex in exactly the same place, and then enzymes that you're familiar with, DNA ligase, to jam them back together again. Okay? That is not exactly how homologous recombination works, but it's a perfectly good way of thinking about it to solve any of the genetic problems that you're going to see for the next year and a half or so. Okay? So just wanted to warn you that that is not exactly right. Now, a couple questions to tease you with is for you should be thinking about what would be the genetic consequence if recombination occurred not between chromosomes of paired homologous, chromatids of paired homologous chromosomes, but between sister chromatids. Tease yourself by trying to figure out, would there be genetic consequences of recombination between sister chromatids? And finally, let's return now to that bizarre orientation of kinetochores on the spindle. I told you before that we had to have tension on the kinetochore for the cells to know that all the chromosomes were attached. How could we have tension here if there's only monopolar attachment of a kinetochore to the spindle? Where could the tension come from? And the answer is up there. Anybody got it? Sister chromatids maintain their cohesion through meiosis I. And so if we break and rejoin those chromatids here, then this pink arm will be connected to that blue arm, which is connected to its sister through cohesion. So the tension on the first meiotic spindle is occurring not through bipolar attachment at the kinetochore, but through the tension that is produced by the sister chromatids being held together on recombinant chromosomes. And that's probably why there has to be one crossover per chromosome arm per meiosis to have faithful segregation of chromosomes in meiosis. Okay? All right, on Monday, we'll discuss the saddest story in the history of biology. Bring a Kleenex, be prepared. And until then, have a good weekend.